Hello, my name is Dr. Sandra Scherer, and I'm a research scientist in Dr. Alana Whelm's lab at the Huntsman Cancer Institute in the Department of Oncological Sciences at the University of Utah. Thank you for joining for this presentation today, which is part of the Jackson Laboratory Humanized Mice Journal Club series. Today, I'd like to share with you the data of our recently published project, which aimed at developing an immune-humanized PDX model of estrogen-independent ER-positive metastatic breast cancer. This was a collaborative effort, and the paper is now published in Breast Cancer Research. Before talking about the results and data published in this paper, I wanted to give a brief background for the project and to explain why we developed this model and why we think it was important and useful to the research community. 1 in 8 women will be diagnosed with breast cancer within their lifetime, and breast cancer is the second leading cause for cancer-related deaths in women. In the US alone, over 40,000 patients die from the disease every year, and this number is rising every year. Cancer cells can become invasive and disseminate throughout the body to form metastases in distant sites or distant organs. And metastasis is the cause of over 90% of all cancer-related deaths. In breast cancer, 25 to 30% of all patients will develop metastatic disease. And at this point, the 5-year survival rate will drop to a devastating 28%. And to this date, unfortunately, metastatic breast cancer is incurable. There are 3 major clinical subtypes of breast cancer: hormone or estrogen progesterone receptor positive, HER2 positive or amplified, and triple-negative breast cancer, which is defined by the lack of the expression of the before-mentioned receptors. Estrogen receptor or ER positive breast cancers are the most common subtypes of breast cancer, and more patients die from ER-positive breast cancer than from any other subtype. ER-positive breast cancers can be treated with hormone therapy, but we know that many tumors acquire resistance to this treatment, and at this point, these tumors can grow back very aggressively, and the effective treatment options for these patients are then very limited to non-existent. So there is a very urgent need for new models that represent the human disease and that can be used to test the efficacy of novel drugs. Current models and existing models of ER-positive metastatic breast cancer include human breast cancer cell lines with its known limitations as a 2D cell line. And one of the major problems here is that the transformed cell lines are not able to resemble the patient's tumor very well. Our labs and others have tackled this problem in a way and established PDX or patient-derived xenograft models to overcome the shortcomings of 2D cell lines. And these PDX tumors have been shown to recapitulate the patient's disease well. But in order to grow human tumors in mice, immunocompromised strains have to be used, which also means that those models lack a functional immune system. And given the known importance of immune cells in disease progression in therapy response, we set out to develop an immune-humanized model of estrogen-independent ER-positive metastatic breast cancer. To accomplish this goal, we decided to use a mouse strain called NSG SGM3 because they're engineered to express the human cytokines CSF, GM-CSF, and IL-3. And they're known to promote a good myeloid reconstitution. So we chose the PDX line HCI 13, which was developed in our lab from a metastatic breast cancer patient sample. And importantly, it harbors a naturally occurring ESR1 mutation named Y537S. And this is one of the major known mutations in the estrogen receptor gene, and it plays a critical role in endocrine resistance. Estrogen receptor-positive tumors are notoriously hard to grow in mice, and they need exogenous estrogen as a supplement. And using our published method, we supplemented the PDX line HCI 13 with estrogen by implanting E2 pellets into mice at the same time of tumor graftment, and then switching to estrogen-containing drinking water 4 weeks post-surgery. The tumor growth curves shown here show the known heterogeneous tumor growth of HCI 13, but most tumors grew within the first 8 weeks. And since most human immune cells in humanized mice develop around 8 or 12 weeks post-humanization, we decided to optimize this method further for immune humanization experiments. We tested both the estrogen supplementation methods side by side alone, and we found that supplementing with estrogen water alone gives us a good window for immune humanization, as seen in the dark gray tumor growth curves. We then tested the effect of estrogen supplementation on immune-humanized mice. And here is a quick schedule for our humanization approach in general. So as I mentioned, we use SGM3 mice and treat them. We then treat them with 2 doses of busulfan, which is a chemotherapy, to induce myeloablation in these mice. 5 days after, we then intravenously inject human CD34 positive hematopoietic stem cells, and those are obtained from donor bone marrows. The following day, we implant PDX tumor chunks and start supplementing with estrogen. At the endpoint, we then harvest tissue. We harvest blood, bone marrow, and spleens, and then we evaluate immune humanization using flow cytometry. In this first experiment, we found that 14 weeks post-humanization, all mice were successfully humanized, given that they had over 25% of human CD45 positive cells or leukocytes in their blood. We also found human leukocytes in the spleen and, to a larger degree, in the bone marrow. We also did further subtyping using flow cytometry that revealed that we were able to detect myeloid cells, T cells, and NK cells, and also some B cells to various degrees in different organs. But to our surprise, we found that exogenous estrogen causes severe anemia in humanized mice, which posed a big problem to our approach. So no comparable phenotype was observed in humanized mice, which were not subjected to exogenous estrogen, or in non-humanized control mice that did receive estrogen. We've also ruled out a donor-dependent effect by testing HSCs from different donors with the same result. Given that the ESR1 mutation Y357S in HCI 13, we investigated if this specific PDX line could potentially grow independent of exogenous estrogen. So for this, we repeated this experiment without estrogen drinking water supplementation, and we saw good humanization again, but unfortunately, we weren't able to detect tumor growth, even after more than 18 weeks after implantation. We then decided to take a different turn, starting from the HCI 13 PDX line through re-implanting into ovarectomized mice. We were able to develop an estrogen-independent subline named HCI 13EI. IHC analysis showed no differences in hormone receptor expression and morphology between the EI line and the parental PDX line. And the fact that we were able to develop this line without exogenous estrogen is consistent with the observation that the patient's original tumor was resistant to multiple lines of endocrine therapy. And importantly, both the HCI13 and estrogen-independent sublines still harbor the heterozygous ESR1 mutation Y537S, which was also seen in the patient's original tumor. When we then checked for endocrine resistance in the HCI13EI subline by treating with fulvestrant. In the parental HCI13 tumors, treatment here in this experiment led to immediate control of tumor growth, as you can see in the upper plot, but without a complete response. On the other hand, the estrogen-independent tumors shown in the lower plot did not initially respond, but show a delayed regression. Long-term treatment of over 300 days were able to control tumor growth, but without achieving a complete response. We then looked into these tumors further, and IHC analysis showed that the residual tumors identified by the expression of human-specific cytokeratin staining had low ER levels following the long-term treatments. To immune-humanize the new HCI13EI PDX line, we then treated mice with busulfan for myeloablation, reconstituted the immune system by injecting HSCs, and engrafted HCI13EI tumors the following day. So flow endpoint data in this experiment showed high levels of human leukocytes in blood, bone marrow, and spleen. And characterization of the immune subtypes showed that at the time of harvest, human myeloid cells were predominant, but we also detected human T, B, and NK cells. The tumor take rate in this experiment was 80% in humanized mice and 60% in non-humanized control mice. We then further characterized the tumors in the immune-humanized mice, and we've double IHC stains revealed that double IHC stains of human-specific cytokeratins and proliferation markers revealed that the fraction of proliferating tumor cells was similar in humanized and in non-humanized mice. We then further characterized the tumors, and IHC stains showed that ER expression is maintained in the humanized tumors, and we were able to detect human immune cell infiltration into the tumors as shown here by human CD4 IHC. Double immunofluorescence stains revealed that a large portion of tumor-infiltrating human immune cells are of myeloid lineage, as here shown by the markers CD11b and CD45. So in summary, our model recapitulates the lymphocyte low and myeloid-rich tumor microenvironment that many ER-positive breast cancers display. For more details on this work, I'd like to refer to the paper that's been published. And in the last minute, I'd like to share one more piece of data with you that might be of interest for this audience. I've mentioned that our model showed myeloid cells that as a predominant cell type in the tumor microenvironment, but we also found a way that we can tweak the immune cell development in these mice. And for this, we used AAVs, or adeno-associated viruses. And in this experiment, those viruses encoded the genes for human TPO, IL-7 and IL-15. Those factors are known to be important for NK cell development. And we were able to screw immune cell development from a predominantly myeloid lineage to an NK cell reconstitution, as you can see here in these graphs on the right. And with this, I'd like to finish my presentation by thanking everybody in the lab who was involved in this project, specifically Dr. Alana Whelm, and Dr. Alessandra Riccio, and Mahi Fujita. I want to thank our great collaborators and funding sources, and importantly, the patients and their families. I'd also like to thank you for your attention and your interest in this project and in this topic. And please don't hesitate to reach out to Jack's Lab or to me directly using this email address posted here if you have any questions about this project. Thank you very much.